Okay, I think these are I think last tool that you guys have to remember. It's called CRISPR-Cas9. I'm not gonna go over the steps for this. The only thing I want you to remember for this, it's used for editing the DNA. They can modify the DNA. So what they did, people before they had this CRISPR-Cas9, they so let's say they were trying to have different species for the plants. One way, that you can get different proteins expressed by the plant or even different species if you change enough nucleotides is through mutations. Mutations can happen normally, or mutations can happen through radiation, chemicals. So people, they started using radiation to cause mutations. One thing with that is you cannot control where the mutation happens and how many mutations gonna happen. Is it really going to give you a good plant after? So then they have this CRISPR-Cas9. Now they don't have to use radiation to cause mutations. They can just edit the DNA. They can cut the DNA. They can the um, uh, add the R. In this case, they're using RNA, guide RNA, and they can turn off. They can modify the sequence with the new nucleotides, or they can remove the sequence. So again, the only thing I want you to remember for CRISPR-Cas9, it's used for editing or modifying. You can add new nucleotides, you can remove nucleotides. So that's CRISPR-Cas9. So that's all I need you to know.
Basically for modification.
For modification, yes. So we talked about some ethical issues. DNA technology in medicine can reduce human suffering. So that's because it can identify diseases. Sometimes it can even improve the chance of living by that. Maybe you won't have the genetic disorder anymore. And it can help parents who cannot have children to have children through IVF and have healthy children because it can scan through PGD and then DNA probing. So that's helpful. And then by detecting diseases early, like Angelina Jolie did for the BRCA gene, they can detect them early, then they can do something about it. And it also gives new treatments for the illnesses that don't have a cure. So if there's no cure, maybe the only thing you can do is you can go and turn the gene off or you can replace it. So that's the only thing that can be done. So that can be done through biotechnology. But then how should it be regulated? Where are the limits for that? I'm going to go over the ethical issues one more time with you guys. Maybe actually you guys go ahead and type in the chat box what, how should it be regulated? What are some of the ethical issues that concern you? I'm pretty sure they did, Giselle. There's a lot of research they do for a mouse. I'm pretty sure they did that even before they did the dolly, the sheep, maybe, or after. So what are some of the ethical issues? I'm gonna call out names if nobody responds. Truth, Frene, Oscar, Devin, Nancy, Audrey. What are some of the ethical issues? Maybe think about one.
One of the ethical issues was a problem with the embryo, an issue, and maybe a parent won't want to go through with it.
So the parents might not have the child anymore. So they can decide to abort or decide to get rid of the embryo. So that's one. One thing I mentioned was the cost. Only rich people can afford it. If they are successful in this and rich people can pick the alleles they want, and they obviously only want healthy alleles, or even going for perfection, they want certain looks, but they can afford it. So if the rich people, because they can afford it, have all the healthy alleles, over time, what's going to happen, natural selection is going to favor that. So the healthy allele is going to stick around longer. they're going to be able to reproduce healthy children if it's done in the embryo. But people who cannot afford it, they're still born with genetic disorders. They might not live long enough to reproduce. So over time, people who cannot afford it might not be able to reproduce, and their population will not be favored by the natural selection. So that's another thing. One thing I think I forgot to mention, let me go back. So here, when they are changing the cystic fibrosis allele, they're replacing the faulty gene with a healthy gene. They're not doing that in the germ cells. Germ cells, I want to remind you again, when I say germ cells, I'm talking about the egg cells and the sperm cells. They're just doing this in the lungs. They're not doing anything with the sperm cells or the egg cells. So this person can still have a child who is born with cystic fibrosis, and then maybe they want to know the percentage. What is the chance they're going to have a child with the cystic fibrosis? Then you can go ahead and do the Punnett square, or the person, the genetic counselor can do the Punnett square and say maybe you have 25% chance you're going to have a child with cystic fibrosis. So what I'm trying to say, this gene doesn't get passed on because it's not directly inserted into the germ cells. Have you guys heard about gene doping? Gene doping? No? What about blood doping? No? Okay, so athletes sometimes, just to get an edge, they do blood doping. That's when they have their blood removed ahead of time before the game. They get the blood. So what happens? Okay, so in your blood there's a protein. It's called erythropoietin, and that's for your body. It gives you ability to inhale more oxygen, and that's going to help you maybe run faster. So when you remove some blood from your body with the erythropoietin, then your body makes more blood, so it reaches the normal range. But right before the game, you have more red blood cells inserted in your body, and now you have more erythropoietin. Maybe you can run longer. So that's, I think I can't remember the name, but somebody did that and they snatched all his medals after they found out. Gene doping, it's when they are going in and they are adding maybe more proteins for that, for even erythropoietin, to make more erythropoietin for your body. So this is, when people take steroids or the drugs that's going to give them edge in the sports, or just to be stronger. So when that's done, it's pretty much the same thing. So they are giving them proteins or the enzymes that will produce more of erythropoietin. So that's called gene doping. People, I don't know if it's legal yet, but there's still research going on. But there is lots of side effects. With the blood doping, it can affect your heart. With the gene doping, it can, your body might not take it up. That's one. But the second thing that can happen is mutations. If there are mutations in your DNA that can cause a disease, it doesn't have to be a positive mutation. It can be totally negative mutation. It can cause frame shift mutation. So there's lots of side effects for that. So there's a question for you guys. Suppose that Jody receives gene therapy for cystic fibrosis via inhaled virus. Could she pass the repaired DNA to her children?